My name is Jamie Brusa. I'm a postdoctoral scholar in the School of Environmental and Forest Sciences at the University of Washington. My co-author, Randi Lupardus, is also a postdoctoral scholar. Our research is called Integrating at-Home Research Experiences with Traditional Laboratories. Undergraduate instructors for science courses currently face the challenge of balancing critical thinking procedures with scientific content. Course-based undergraduate research experiences, or CURES, have become a popular method of instruction because they provide access to research experience for all students. However, implementing a CURE into a course curriculum comes with many challenges, including financial barriers, increased time investment for both instructors and students, incompatibility with limited or no lab time, which could be the case during a pandemic, and a narrow scope of topics and laboratory skills gained compared to traditional laboratories. Ideally, scientific laboratory curricula include learning outcomes for students to gain a diversity of laboratory skills, reinforce scientific concepts, practice higher order thinking, and develop an understanding of the research process. Given the vast amount of information covered in introductory science courses, students might benefit from a laboratory curriculum that focuses at least partly on reinforcing complex concepts discussed in the lecture component. In research-based laboratories, students might struggle to make connections between theoretical concepts discussed in lecture and research experiences in laboratory sessions if the research topic is too narrow. Further, students might feel that they do not have adequate time to focus on their research project during limited laboratory time. This conundrum led us to our research question. Can students benefit from engaging in the research project at home? Our research focuses on assessing the utility of updating a one-semester introductory cellular and molecular biology laboratory curriculum to employ techniques of both traditional laboratories and CURES. We integrated an at-home semester-long research project with weekly themed laboratory experiments such as osmosis, enzyme function, cellular respiration, and photosynthesis. Students chose one of 3 general frameworks provided for their semester-long research project: an experiment or observational study using soil seed banks, an experimental study using green onions in a highly controlled setting, or an observational study of seasonal biological events. Our laboratory curriculum framework capitalizes on the benefits of a CURE and a traditional laboratory by being inclusive to all students to learn research methods, providing inquiry-based instruction, providing opportunities for critical thinking, helping students reinforce lecture concepts, and being time and cost efficient. A semester-long project can also be used to replace labs if such a setting is unavailable in uncertain times, such as during a pandemic. We evaluated our novel curriculum using survey questions and correlational analysis between performance on the research project and overall performance in the course. Our results indicated that students performed their semester research on a variety of topics. We had 71 participants and found that students who performed well on their at-home semester-long research project tended to have high overall course grades. And mean student responses from the survey questions indicated that students agreed strongly that conducting the semester-long research project helped them appreciate the realities of scientific research, such as the importance of a good experimental design and challenges associated with unexpected problems. Using components of CURES in the classroom is an easy way to teach students about the process of scientific research in an authentic manner and is easily adaptable to lecture and laboratory contexts with limited resources required.